Welcome everybody again to another episode of the ideaXme show that profiles the human beings behind the really big ideas that are shaping the world, inspiring the future and future possibilities. And for all those that like really great stories, I'm Ira Pastor, your life science ambassador along for this journey today. Today we are going to head back towards the topic of Alzheimer's disease, which, as we've discussed on previous episodes, is one of those conditions that, while not responsible for, say, as many deaths annually as, say, cancer or heart disease, is a problem that is growing substantially in our rapidly aging population and is predicted to become an epidemic of major proportions in the coming decades. The estimates are all over the place, but the current numbers suggest that we have about close to 6 million Americans alone, most over the age of 65, that have some type of dementia caused by Alzheimer's, estimate about 18 million or so around the world, and those numbers are supposed to go to 14 million in the US, 100 million globally by the year 2060. So this is clearly a disease of substantially unmet medical need. There's no current cure for Alzheimer's disease. Available treatments, as we've discussed in the past, offer quite small symptomatic benefits and are primarily palliative. And the predominant area of clinical research, which is focused on treating what has been thought to have been a primary underlying disease pathology, that is beta amyloid plaque formation, and hence the attempted reduction by small molecule drugs, immunotherapies, and so forth, have been using up billions of dollars in research funding and have yielded quite lackluster results in clinic to date. So much so that many members of Big Pharma are actually getting out of Alzheimer's disease entirely, which is a scary thought. This has prompted a set of new thought leaders to begin to ask the big question, is beta amyloid even the right target we should be focusing on? Or is it, and related tau, tangles, and damage far downstream from much more important biologic events that we should be paying more attention to? On a few previous episodes, we've discussed some of these themes. We've talked about the infectious dimension of Alzheimer's disease in regard to work in the area of chronic herpes infection. We've talked to some folks that have looked at Alzheimer's disease as a new form of diabetes from a more of an endocrine angle. Today, we have a fascinating guest. Dr. Michael Fossel is founder and president of Telocyte, and has recently published a fascinating review in the Journal of the Alzheimer's Association entitled A Unified Model of Dementias and Age-Related Neurodegeneration, which focuses on a theme of a failing class of brain cells becoming senescent, entering a state of irreversible cell cycle arrest, called glia, which are non-neuronal cells in the central nervous system that primarily act to maintain homeostasis and provide support and protection for other neurons. And looking at this as a major driver of Alzheimer's and dementia, it's working on an innovative gene therapy that could ultimately target these cells to ultimately help keep dementia at bay. Dr. Fossel was recently professor of clinical medicine at Michigan State University. He's a respected lecturer, author, and founder, and former editor-in-chief of the Journal of Anti-Aging Medicine, now known as Rejuvenation Research. Dr. Fossel earned both his MD at Stanford and his Ph.D. in neurobiology there. In addition to his position at Michigan State, Dr. Fossel's lectured at the National Institutes of Health, the Smithsonian Institute, and various other universities and institutes around the world. Served on the board of directors of the American Aging Association, was their executive director. And he has published extensively. He's written numerous articles on aging and ethics, as well as co-authored or authored several books, including The Telomerase Revolution, The Immortality Edge, Reversing Human Aging, Cells, Aging, Human Disease. A lot of stuff to talk about. Dr. Fossel, thank you for taking the time to come on the show today.
My pleasure, Ira. Thanks for having me.
Absolutely.
Sounds like fun. You gave a great introduction, not to me, but to the field. You nailed it.
Thank you. Thank you. I tried to do that so you didn't have to lecture for 20 minutes about it.
No, I mean, that's exactly been the problem with Alzheimer's and age-related diseases, and I think you summarized it dead on.
Great. Great. So then moving from that, so we got that stuff out of the way. I want to give you the floor for just a little bit. Typically, we start the show off giving our guests the floor, talking about them, your background, where you grew up, sort of how you got interested in medicine.
If I grew up, right.
I say the same thing about myself. And sort of, along your history of teaching medicine, of, specializing in emergency care and so forth, how you got interested in aging and neurobiology, Alzheimer's, dementias. Take us on a little bit of that path, if you would.
I started off actually in psych, did a BA and an MA in psych. Found I could get it for free because they were charging me by time and not by credit. So I just doubled up the credits and got an extra master's. And then I ended up getting some extraordinary GRE scores, and my advisor talked me into going to graduate school rather than going off learning languages. So I ended up going off to Stanford. And I was teaching neuroanatomy and neurobiology in the med school. So one day they turned to me and they said, you can be a professor or a lecturer. Would you like to go to med school? I said, sure. And they said, lecturer, professor. And I said, well, I'd love to be a professor. Why would I want to be a lecturer? They said, well, lecturers get free tuition. So I did. But what intrigued me, in those days, I was working on the development of the nervous system, how it comes together. And I must say, it absolutely floors me. Just the stunning complexity and the way things get together. It's like staring at the Milky Way galaxy and just marveling at nature. And that was true of development of the nervous system. But I found that when I would talk to people around me, researchers around me, and talk about the way the nervous system came apart in old age and other age-related phenomena, they would sort of shrug in a very blasé way and say, well, things just happen. What do you expect? You get old, things fall apart, you rust. And I've always been a little bit of a contrarian. When people get blasé about things, I suspect there's more to it than that. Whenever somebody says it just happens, I suspect there's a lot more to it than just.
Sure.
But I wasn't sure what it was. In those days, there were a lot of interesting leads. Example, reactive oxygen species. Example, progeria. There were endless kind of fascinating parts that somehow played a role in aging, but it was hard to put them together. So rather than get into the research, I moved into clinical medicine, which I thoroughly enjoyed sort of as an avocation in a sense, rather than a real vocation. It was fun. I enjoyed it. But I was fascinated by aging. So in the early 90s, I decided to write a textbook about this. I found one, a friend of mine who wrote, I think, a better textbook. And two, I ran into some of the early work on cell senescence and telomerase. Sure. And I was fascinated because I began to see how you could begin to put together these pieces of aging and have a unified model. So I ended up taking a look at some of the early, some of it confidential proprietary data from Jared in those days out in California. Sure. And putting a book together on it. And that's why 24 years ago, I gave the first lecture ever in history on reversing human aging at the NIH. One of the things I said was that, anybody who leaves this room in an hour and thinks you can reverse aging is naive. Anyone who leaves this room in an hour and thinks you can't reverse aging is naive. If you're sensible, you'll leave here and say, where's the data? Let me tell you where the data is, because here's what we got. And I think that's still true. And there's a lot more data now that begins to put the model together. But a couple of things hit me. One is, in some sense, I don't care what causes aging, how it works. I want to know where we can intervene effectively to make people's lives better. I don't mean longer. That's fine. I mean better. Make people healthier, better, enjoy themselves. That's what concerns me. For example, Alzheimer's disease. But the other thing that hits me, though, and it's hit me for a long time, is that we tend to take a very sort of simplistic, magical view of aging. And historically, it reminds me of the same sort of thing that happened if we go back to the Middle Ages and ask healers in those days what caused plague, for example. And there were people who thought it was caused by eating fish. People who thought it was caused by witchcraft and curses and God's will, lots of ideas. But what struck me was that what their approach to plague, for example, or smallpox, is in many ways parallel to our approach to age-related diseases. There's a quote in my recent paper we'll come back to, where Len Hayflick says, the problem with age-related diseases, everybody agrees aging plays a key role in age-related diseases, then they entirely ignore aging and concentrate on the disease, namely the signs and symptoms. So if we went back 500 years, a lot of times we have a tendency to look back in history and think people in the old days were dumber than we are. No, I don't think so. They had different conceptual ideas about how the world worked, but they were bright people. And I will bet you that if we went back 500 years, 600 years to medieval Europe, we would find that the herbalists, for example, were incredibly bright, incredibly knowledgeable people, far more than I will ever be about herbal medicine. But they really believed that the treatment, for example, for smallpox or bubonic plague was to use herbs. And you know what? Good tool, wrong target. Doesn't work. What you really needed to do, for example, with regard to smallpox, is understand that it's caused by little teeny invisible things you cannot see with your naked eye. Sure. And it's microbes. It's virus, bacteria, for example, with regard to plague, bubonic plague. It's a bacteria. And until you make that conceptual jump, I don't care how good you are at herbal medicine, small molecular approaches, you will fail. That's kind of what's going on with regard to Alzheimer's and other age-related diseases these days. We tend to think that the way to treat, for example, age-related diseases is by using specific small molecules, NAD+, any number of things, resveratrol. But we're looking at it still with the wrong framework. Instead of looking at the whole system, we're looking at components. We're looking at biomarkers, hallmarks, components, pieces, rather than the holistic system, and rather than saying, how does it work? How does the process work? Here's an example. We know that aging is one of the major risk factors for Alzheimer's, and yet I know 45-year-olds in our registry who have Alzheimer's, younger than I am. In fact, 35% of the people in my registry are younger than I am. I know 100-year-old people have no evidence of Alzheimer's. Now, that's not to say that aging doesn't play a role, but you can't simply say aging causes Alzheimer's. The verb cause is a misnomer. Same thing is true of genes. I know that if I had 2 ApoE4 alleles, which thank God I don't, my risk for Alzheimer's goes up enormously depending on that age. And yet I know people with 2 ApoE4 alleles who have no Alzheimer's, and people with an ApoE2 and ApoE3 allele who have Alzheimer's. So again, that's not to say that genes don't play a role. They do, but it's not causal. And the same goes for head injuries, for example. Same goes for exposure to radiation. Same goes for hypertension, diabetes, various infectious disease, bacteria, fungal, chlamydial, even prions, standard prisoners. All of these things have been implicated as increasing your risk. And yet to think that they cause Alzheimer's is a misnomer. And the same is true at the bottom end. You alluded to beta amyloid, which has been sort of the key model for the past 20 years, call it. And it's been around for 110 years, but key model in terms of intervention for disease-modifying therapies for 2 decades. And yet, every time you intervene in it, you don't have a good effect. So whether you're looking upstream at risk factors or downstream at biomarkers, what you find is the model's still wrong. It's like me, again, going to something like Ebola or rabies or smallpox and saying, I noticed that, if you are around, if you're older, you have a higher risk, for example, of coronavirus. So that must be the cause. Or saying that downstream, if you have a fever, that must be the disease. Well, when I'm looking at things like typical viral infection, fever, vomiting, maybe bleeding in the case of Ebola, nausea, a high white count, those are not Ebola. They are parts of Ebola. They are components, but they are not the disease. And the same thing is true of Alzheimer's. Alzheimer's is not beta amyloid, is not tau tangles. They are components of it, but they're not the process. And what we tend to do is we look at things like the genes upstream, the markers, biomarker of tau tangles and beta amyloid downstream, and we think of those as the disease. And the problem is they're not. They're components, they're parts of it, but they're not the process. The other thing I find a problem sometimes, people talk about causation. And causation is great when you're dealing with classical physics, but it tends to fall apart in quantum physics, for example. And I think the same is true when we get a really complex disease or other biological phenomena. For example, what if I said, what causes evolution? Well, that's it's not easy to answer that in one sentence.
Nope.
I don't mean that there isn't some cause and effect in things that happen. I don't mean that at all. I just mean it's so complex that to simply use the word cause is to sort of narrow your thinking too much. Sure. For example, genes play a key role in evolution, but genes don't cause evolution. They are a key part of evolution. The same thing is true is when we're getting to Alzheimer's disease or any other dementias. To say that, for example, telomeres cause aging, they don't, but they're a key component. Cell senescence causes aging. No, it doesn't, but it's a key component. Beta amyloid or alpha-synuclein in Parkinson's disease, and upstream genetic factors. They don't cause it, but they are key players. They are components of this holistic system, this dynamic system that we need to understand. And from my perspective, as I've said, in some sense, I don't care. What I want to know is where is the single most effective point of intervention? It is, and financially for that matter, it is clearly not just lowering your blood pressure, controlling your blood sugar, avoiding radiation, wearing a helmet when you do sport. That's clearly not good enough. It's good, it's just not good enough. Clearly, likewise, downstream, it is not simply a matter of dealing with beta amyloid or tau tangles. Good idea, not efficient enough. The question is there a better point of intervention? Yeah, I think there is, which is why I wrote the paper, which we'll come to in a minute, I suppose, too.
Absolutely, and I really appreciate your overview in that context. I think the principles that we too often don't think about in the world of systems biology and how all these pieces play together is so intrinsically important to this discussion. And I think I really like to start off there in the context of this perspective on glia and more this principle of the looking at the brain as this microenvironment of, the brain contains many different types of cells doing many different things. We talk about the glia, you usually don't think about the central nervous system and the brain as having substantial turnover, but here's a class of cells that does indeed. It's susceptible to senescence, telomere shortening, and so forth. Let's start out there. Talk a little bit about glia, their role in sort of keeping the whole system, or helping out, let's say, keeping this system normally in check and healthy from the point we're born to later on, and why it became such an interesting domain for you to focus on.
Tough question to answer specifically. Glia are the majority of cells in your brain.
Mm-hmm.
We tend to focus on neurons, and part of the problem is it's a little easier to identify what neurons do than it is to identify what glial cells do.
Mm-hmm.
That doesn't mean glial cells are in any sense less important. It just
Right.
means we have a hard time wrapping ourselves conceptually around the function of glial versus neurons.
Yep.
When it comes to things like oligodendrocytes, which are the glial cells that wrap themselves around the axons, that's
Mm-hmm.
a little easier, and we make fairly vague, actually, noises about how they function as insulation. Fair enough, and not wrong. I suspect there's, again, more to it than that, and there
Sure.
is. And the glial cells, they're originally named from the Italian word for glue, glia. Ramon Cajal, a long time back, remarkable guy. But the thought was they sort of glue all the neurons together, and I think for a long time, without thinking about it, we thought that glia were sort of just the background. It reminds me, by the way, of our view of junk DNA. Namely, we look at, say, 2% or so of the genes, and we say we know what those do. We have an idea they express proteins. The rest of them are junk. Well, no, they're not. They clearly are more than that. In fact, they're probably predominantly regulatory and very key players in how we survive and how our cells work. But the same is true of glia. They are certainly not junk. They're not just glue and background things. They're not just structures on which the neurons sit.
Sure.
And we do know that they play a lot, among other things, in support. I don't mean physical, I mean metabolic and physiologic support for the neuron.
Right.
It's particularly around things like synaptic function, but also in general around neuronal function. Example, beta amyloid. One of the things that glial do is that they produce beta amyloid, they then bind the beta amyloid, internalize the beta amyloid, degrade the beta amyloid, make it again.
Mm-hmm.
And that brings up a key issue that always comes up that people miss, and it pertains to glial cells too. People have this idea that your molecules just sit there. Example, if I'm looking at wrinkles in my skin, people will think somehow that you were born with a certain amount of collagen and elastin, and over time it breaks down. It just happens. No, it's a dynamic population. It's a dynamic pool. And when you're young, you're continually turning over, for example, elastin and collagen. When you're older, the turnover rate's slower. The same is true of beta amyloid. If you look at cells as they become senescent, you find their turnover rate slows down. Well, the outcome of that is sort of like turning down the maintenance rate in your house. If you take care of your house once a week, it looks pretty good. If you take care of your house once every decade, it doesn't look so good. And that's kind of what's going on. It's the rate of turnover that has an enormous impact on the function of the cells around it, in this case, neurons, which tend to first lose synaptic ability and then actually become dysfunctional and die. And you and I are left with Alzheimer's. By the way, it's not that simple either, but that's a good place to start.
Yeah, no, that's why I wanted to start off there. Now, so stepping down from there, obviously, you've written a lot about the telomeres and telomerase. We haven't done a telomerase-centric show, but we've discussed a bit of these topics, the importance of these the telomeres in the sense of these genetic caps on the ends of our chromosomes that play these very critical roles, cell cycle regulation, aging, cancer, and so forth. We talked a little about the history in the past in terms of Blackburn and Greider and Shostak, getting the Nobel Prize for the 1980s discovery. But we really focused too much on sort of what went on in the 1990s, where you had some initial small molecule pharmacotherapeutic interventions towards telomerase as a target, primarily on the cancer side, Geron J&J. There was a little activity on the reverse of that. Could you just sort of walk through a little bit of the pharmacotherapeutic history of telomeres and telomerase, which wasn't that, I know they had great outcomes, but then ultimately, parallel to that, we had sort of the gene therapy realm. Once again, ups and downs, but now we're at a place where things are looking a little better. Just talk a little bit about those two paths as they were occurring in the past, and ultimately bringing where we are today, because I think this feeds in nicely to ultimately what you're attempting to do.
People have this feeling that, and it's inaccurate, that science is sort of progressive and reasonable. Science, like women's fashions, comes and goes.
Sure.
Comes in waves, comes in fashions. There are any number of irrational things that occur in science. And that's certainly been true with regard to telomeres, for example.
Yep, absolutely.
There are times when it was in vogue 24 years ago. There are times when it's coming back into vogue, maybe now. There are some irrational ideas where things are overly simplified. Example, the idea that extending telomeres causes cancer. No. And I understand where it came from. Judy Campese's old remark about the double-edged sword. And it's not entirely wrong, but it is simplistic and doesn't reflect reality very well. So science has had this problem, and particularly in this area, for a long time. As you probably know, the first person ever defined, well, the idea that there were such things as telomeres goes back about 100 years. But the first person who defined the need for there being something called telomerase was a friend of mine, Alexei Lovnikov, who's still alive and kicking in Moscow. Interesting guy, one of my favorite crazy people. But he said, there has to be a thing called telomerase, and we gave good reasons for it, because otherwise there wouldn't be life on this planet, at least not eukaryotic life.
Right.
He was right. And then along comes another friend of mine, Len Hayflick, who in 61, along with Moorhead, pointed out that in fact cells do age. Aging is not simply a matter of the gestalt, it's a matter of cells themselves show these changes. And he talked about a thing called a replicometer, which later on was shown very nicely in the 90s to be telomeres. That is, as telomeres shorten, by the way, it's not the length of the telomere, it's the relative length of the telomere. We'll come back. As that occurs, you see these changes occurring in cells in terms of cell function and gene expression. Now, the question that was present in the mid-90s and really got settled in 1998, now 22 years ago with a Bodnar paper, was that correlational or causal? And what the Bodnar paper did was show that if you re-extend telomeres, you reset the cell's lifespan. And in fact, you reset gene expression. So if anybody ever said, geez, will we ever reverse aging? Well, yeah, we did that 22 years ago in human cells. We showed you can reverse aging. Works very nicely, thank you. I don't mean we're great at it. I mean, it can be done. And then if somebody ever said, could we do that in human tissues? That was shown now 20 years ago this year in human skin, and 19 years ago this year in bone, followed by arterial tissue. So we know that we can, for example, take old arterial tissue that's pathological, shows coronary artery disease, and use those cells, reset telomere length, reset gene expression, and grow young arterial tissue. But you and I aren't just a couple of cells in a petri dish.
No.
We're certainly more than that. And then Geron was playing a prominent role in a lot of this research in the 90s. Geron sort of had three platforms. They had stem cells, which Mike West took off and is still running with.
Right.
They had this whole program with telomerase, and the idea here would be the kind of thing I just talked about, resetting aging in cells, tissues, and ultimately organisms. And they had cancer, that is anti-telomerase. And they elected to sell the first two and keep the last one, which is probably why they still don't have a product now, quarter century later, which is unfortunate. And then, I got involved in this. I know back in around 2001, I was offered an enormous amount of money, $1 billion actually, to take this all to translational work. And that fell through because of a marital spat, where somebody decided not to do this after all, which confused me for a couple of years. But it was probably just as well. We didn't have the technical ability to do what we would have liked to have done back in 2001. Right. So I ended up writing a textbook about this. It's still the only textbook on the topic. It is completely unreadable. It's written in high academic prose. Good luck. But then, about 5 or 6 years ago, it must have been about maybe 8 years ago, 7 years ago, somebody wanted to know if I would join their biotech company and see if we could take some of this work to treating Alzheimer's in those days using a different approach. And then they asked me to write a book about it, sort of to help, which is where the Telomerase Revolution book came from, which the Wall Street Journal said was one of the best 5 science books of the year. Good, but it didn't pay the mortgage. Now, as with many biotech companies, it wasn't bad science, it was bad people in a sense, some personal problems involved that let that fall apart. Often true with any business. Yeah. And then about 3 or 4 years ago, a friend of mine now came from the UK to come see me and wanted to start Telocyte. So we did, because we're going to go ahead and take this and show that we can not only cure Alzheimer's, but other age-related dementias, and probably a good deal more. So that's sort of the thumbnail of it. What did I skip about your question, Ira?
No, I was that's a great history on the pharmacotherapeutic stuff that's going on. Talk about the gene therapy evolution alongside of that, because we went in.
You know, we have had, theoretically, we've had the idea of of course, gene therapy in the sense of breeding animals have been around as long as human beings can think about it. But the idea of active gene therapy conceptually has been around for a good number of decades. We can trace it back probably into the 60s. But as an actual intervention in clinical trials, it's been around for just barely longer than two decades. And there were some initial positive results, but then there was one big problem with the Jesse Gelsinger case, where he died. Nor was that the only one, but there were some missteps. And as ever, you know, it wasn't just the science, partly it was just humans being humans. And we make mistakes, so do I. But the result of that was to sort of put the kibosh on gene therapy in a lot of ways for a good decade in many ways, depending how you measure this. But it certainly put the brakes on in many ways. And it's come back into its own. One of the people who was on our board was the chief scientific officer for Avexis, which was the first group that showed that you could literally cure spinal muscular atrophy using Zolgensma. They actually got bought by Novartis for $8.7 billion, which left a lot of eyebrows on the ceiling as that happened. But you know, we're beginning to make inroads on this in a number of ways. You know, I think it's about 5 different sort of genetics approaches that are fascinating. One is to put in a new gene. One is to simply go, not simply, if you put in a new gene, you can instead try to rewrite the gene you have using, for example, CRISPR-Cas9, or you could take out some cells and then use CAR-T to alter the genes and put them back. And another approach is the one that's being used in some infectious disease ideas, which is to use viral particles to go after some bacterial diseases, that is, use bacteriophages, and alter them genetically so that they're really good. Instead of having an antibiotic, you can actually tailor a virus to go kill, for example, E. coli. And the 5th approach is the one we're taking, which is to actually put in a perfectly normal gene, in this case, a gene for telomerase, and re-extend telomeres to reset gene expression. We are not interested in whether it's still expressed months later, as most people would have. If I'm trying to treat your sickle cell, I want it to last more than a few months. In our case, we just simply want to reset telomere lengths, reset gene expression, and have cells behave functional again. So that's 5 different genetics approaches that are currently out there in one form or another.
And so here we are, we're in 2020. Telomerase remains a viable target. Gene therapy has evolved in its own way. You're now Telocyte, you're putting the two together. If that, whoever was having the marital spat back in the day showed up tomorrow with the $1 billion, obviously nothing confidential here, but what's a little bit of your play? Where are you taking this? Because it's fascinating, but I love to ask the translational question because I want to see as much of this stuff get done. I mean, first caveats on the $1 billion, but you get the $1 billion now.
Well, I mean, first the humorous remark about that, which is given the track record of these people in the past, the first thing I do would be to say, I don't want to hear it. Talk to my lawyer. When the thing's signed, the money in the bank, come back to me.
If you found $1 billion in a cab tomorrow, what you?
No, I mean, because they flew me out to California, and you know, we spent 6 months together putting this thing together before the lawyer called and said, we're going to pay you a lot of money not to sue us, but we're not going to tell you what happened. Okay, I didn't know. I thought it was me. No, it was their marriage. Okay, But I sometimes think of this in terms of rate limiting steps. So first I'll give you the overview, and then I'll talk about the rate limiting steps.
Mm-hmm.
The overview is, I think that we can demonstrate an effective cure for Alzheimer's in 24 months for $10.3 million.
Okay.
That's the quick answer. Now the slower answer. What are the rate limiting steps? That is, what keeps you from doing it now?
Mm-hmm.
The first thing is funding.
Sure.
This is one of those things I call the chasm problem. If I'm faced with a big chasm, I can't jump it in two jumps. I either jump the chasm or I don't. I can't jump halfway and then middle of the chasm falling down and jump another.
Sure.
And that actually turns out to be true with regard to putting together the gene therapy we need. As I've sometimes said, for me to treat 12 patients with Alzheimer's cost about $4 million. For me to treat 140 patients, real number, cost $4 million. For me to treat one patient cost $4 million. Yep. It's the setup costs and the quality assurance, making sure that this is done with absolute quality, so the FDA agrees. It's done with safety, it's done with efficacy. We can't shortchange that. So that's the price sort of no matter what we do. And then price after that falls dramatically. So the first thing we need is funding. And again, we've got funding to sort of pay the bills, but not enough to jump the chasm. That's what we need. The second rate limiting step is the FDA, and that's not much of one. I've talked to them informally. They're very enthusiastic and supportive. They're not going to let us cut any corners, but they'd like to see us take this to clinical testing too. I find that the FDA are very careful people, not unreasonable people, but they are careful, and I appreciate that. With my history in medicine, I don't want people who are just thoughtless and uncaring. I want safety, I want efficacy, so do they. So again, I'm with them on this. The third actual rate limiting step, curiously enough, is the production. I can probably get this produced very nicely by a college campus half an hour from here. I can do it in Mexico, I can do it in Colombia, I can do it in any number of places. Mm-hmm. But if I'm going to meet FDA standards and get the quantity I need, the quality I need, the safety I need, I have to do it right. If I go to the FDA, typically about 2/3 of the attention is not spent on the patient issues, it's spent on the production quality issues, CMC issues, I call it. They want to make sure that when you do this, you really are producing what you think with safety, with reliability, with validity. It has to be done right. So to do that, to get the production I need with quality I need, takes me about 6 to 8 months minimum. Sort of the way it is. So what we're looking at is the funding, going to the FDA, starting the production. Then there's about a 6-month period where I have to get the animal toxicity study done, required by the FDA. And then after that, they give me an IND and I start the human trial, which runs me about 6 months too. Now, in between there are times when you're looking at the data and there's setup times. But roughly speaking, if all went well, it never goes all that well, you're looking at 24 months minimum.
And you have $990 million left over to do other things, which is good.
Well, yeah, I mean, you were right. We're talking about, for example, Biogen Eisai, as of middle of last year, has spent $750 million, just short of that, $747 million at one point, on a failed study with the anakinumab. And they're still trying to rewrite it. I was there when Samantha Heberlein was trying to explain this a month and a half ago in San Diego. And I like her. She's very professional. She's incredibly knowledgeable, but no one quite believes the cherry picking of their data. And the global consensus is anakinumab either doesn't work or works so minimally that that's nice.
Sure.
Certainly not something you want to spend $750 million for. No, I think we can do it for a lot less, a lot faster. It's just a matter of picking the right target, not going downstream, not going upstream, but saying, where do we get in here that'll give us the biggest bang for the buck? And I think that point is in this case, telomeres. It's not because telomeres cause aging or telomeres cause Alzheimer's. They don't. And any more than, as I say, genes don't cause evolution. It's a question of where you can apply the maximum impact.
All right.
It's the optimal point of intervention clinically and financially. That's probably telomeres. Just part of the process.
Yeah. I think, look, I think in 2020, you have the stars, at least, putting the funding aside for a minute, the stars have aligned in the sense you have a decent target, you have the evolution of gene therapy, and you have regulations that, which probably didn't exist back in the day for, these living therapeutic substances. FDA has them, EMA has them, that, basically gives you a path. This is how you develop this thing that looks a little different than a traditional drug or a protein, which is, I think everything is lining up. So I think that the time is wonderful for it.
Well, yes and no. I mean, there's still limitations which we can come back
Sure.
to, things that badly need to be improved. We're nowhere near where we need to be. And right now, I can go ahead and administer this to you, and I can probably get less than 1% of your microglia. I can probably get something in the order of 1% of your astroglial cells, and I can probably get something in the order of 5% of your neurons. Well, boy, that's a small amount. Well, on the other hand, it works when we try it. We do this on animals, the same result comes out very nicely. We can do better. The question is, can we get a better viral vector? You know, when I was at the Gene Therapy for Neurologic Disease conference last month, you know, the holy grail in some sense for neurologic disease is finding vectors that go after microglia. Everybody would like that. We'll get there. What we've got, I think, is good enough to demonstrate that we can probably cure and prevent Alzheimer's disease. It is not as good as it needs to be.
Well, that's right. I wanted to go a little further on that one because I think it's very important. Because I had Tony Atala on the show a few months ago, and he always talks about sort of, you know, you can do a lot with your organs when they're breaking. I mean, it takes a lot of organ degeneration before your organ fails. And I think, you know, I think about this in the same context we were talking about. You know, how much do you really need, though, in the initial context? You think these gene therapy products are going to be improving 20, 30, 20, whatever, we're going out. Still, you could probably get a decent, as you were saying, result, a therapeutic outcome that is acceptable, even with those small numbers, you know, because it's not all about, you know, it's not your entire brain. There's components of your brain that ultimately start breaking down through this process. It doesn't, you know, just snap your fingers, it happens instantly. I'm very interested in that. And then also from the perspective, when you move, obviously you write a lot about aging. When you move into the broader topic of systemic aging throughout the rest of our body, are there tissues that you are thinking about, like the glia in the brain, that, you know, maybe that useful support tissue to start with, as opposed to, you know, when you think about some of, and we can talk about this later, a lot of the telomerase gene therapy stuff that's happening outside the FDA now. Does it need to affect your whole body? Are there specific niches that may be more useful than others, where even if you only got, you know, a couple % of these cells, it's going to have that trickle down effect to others? Talk us through a little bit of that, because I think it's really important in the context of how much you need to ultimately impact in the short term.
Yeah, it's funny how many different things play a role in this. For example, if I'm going after the brain, one of the issues is, can I get it into the brain?
Right.
Blood-brain barrier, for example. One of the issues I just alluded to is using the viral vector to deliver this. Do you get the cells you want to, and how much of them can you get, and how long does it last, and so on. But
right.
It gets remarkably complicated. I mean, it sounds good to just say you can do this. Yes, but it's not quite as easy as that. Technically, it takes some real effort and thought and care. But when you're dealing with the rest of the body, one of the things that comes up are strategic issues. We have chosen to go after Alzheimer's. And you could easily say, why not Parkinson's? And the answer is, well, Alzheimer's tends to be a scarier thing to most people, and there are more patients globally at the moment, and there are a lot of strategic reasons.
Right.
You could also say something at the other end. You could say, why don't you go after wrinkles? There's certainly a big market for anti-wrinkle therapy. On the other hand, nobody ever died of wrinkles.
Yeah.
Okay? I'll give you a slightly more practical example: osteoporosis. No one ever dies of osteoporosis. Now, they do die as a result of it, but they don't die directly because of it. You know, you're 87, you've got bad osteoporosis, you slip, you fall, you break your hip, you go to the hospital, you end up with a little pneumonia. The next thing you know, it develops, it's worse, you're in the ICU, now you've got urosepsis, and then you're dying.
Right.
But it wasn't the weakening of the bones that killed you, it was all the complications. Okay. There are other treatments, whether they're good or bad, bisphosphonates, calcium, and, you know, we can go on and on about things that sort of have some impact on osteoporosis clinically. But if I come to you and I say, Ira, you've got pretty bad osteoporosis, we got some alternate therapies, and it's not going to kill you, but we have this great gene therapy, it's experimental, we're not sure what it's going to do, but we'd like to try it. Your appropriate answer, and so would the FDA's, be, you know, run some tests, do it in animals, try some early studies, get back to me and talk to me in 5 years and let me know, okay? Right. And that's appropriate. But if I come to you with Alzheimer's and I say, listen, you definitely have Alzheimer's, your mean lifespan's 8 years, some people a lot less, some people a little longer. Global consensus is that there is no therapy that's effective in any way. Never mind some people's claims. The global consensus is, nope, isn't there. And I say, you know, I've got an experiment. And you say yes. You don't even want to hear about the possibility. You know, if I'm talking about experimental gene therapy in the context of Alzheimer's, most people say, you know, anything, I'll try it. And the FDA, to some extent, has that same feeling because it's a sort of risk-benefit ratio. I'm looking at osteoporosis, not so much. I'm looking at Alzheimer's, a little more leeway. It's not that we intend to be unsafe and harm people, but you know, if I said you have a 1 in 10 chance of dying of a therapy, which you don't probably from this, and you have a 100% chance of dying of Alzheimer's without it, I'll go for the 1 in 10. I'm not even interested so much in the side effects. And I don't mean to overplay this, but strategically, in terms of patients, ethics, regulatory burdens, it's a lot easier for us to go after Alzheimer's than it is to go after osteoporosis or osteoarthritis, or even if we're looking at the major killer globally, really, which turns out to be age-related arterial or vessel disease, that is, vascular disease, not just arterial, venous too, capillary too, but arterial, particularly things like strokes, heart attacks, and so on. Because if I come to you and say, you've got pretty severe arterial disease, age-related arterial disease, you may, in fact, live another 30 years, or you might have a stroke or heart attack tomorrow. Furthermore, there are other treatments. Some of them are very expensive and uncomfortable, cardiac transplant. Some of them are pretty cheap, statins. Some of them are pretty easy lifestyle choices, change your diet, exercise. But the point is, you have options and you don't know that's going to kill you. Alzheimer's is not that way. I think that our biggest market globally and our biggest opportunity for patients to improve lives is to go after age-related arterial disease, followed by osteoarthritis, followed by osteoporosis, followed by age-related renal failure and podocyte senescence. There are a lot of options.
Sure.
But strategically, and for a number of reasons, Alzheimer's is our best target for now, followed by dementias generally, hence the paper, followed by other targets.
Yep. And it's a big one.
Yeah.
I mean,
we talk about aging and longevity, and you go to anti-aging conferences. This is not talking about extending the lifespan. This is talking about making people healthier.
Absolutely.
Incidentally, you'll probably live longer, but while you do it, you'll be playing tennis, wrestling with your grandchildren, and cooking, or wandering the world. Yes, good idea. And not being sick. Thank you.
Yeah. When you published the current paper, the unified model, you wrote an epilogue, which I thought was fascinating, where you talked about all the reviewers and all these alternative opinions that they threw at you during the peer review process, which I thought was kind of rare.
Yeah, it was.
Do you think, I mean, it's really fascinating, but you see it all. I mean, I spent my entire career in the pharmaceutical industry, and I know sort of the battles that went back and forth between cardiology and oncology and so forth. What's your current take? I mean, what's your current feeling in terms of let's say the institutional narrative, let's say, around Alzheimer's? Is it moving at all? Because a lot of the stuff in this epilogue was the typical stuff, right? Do you look at this, that, the other thing? But this, that, and the other thing was all the old stuff. It's that typical, they don't want to get you off the target too much, but you're going in a much different direction that's potentially a breakthrough in the context. Was this a difficult paper I don't know how many iterations it took for you to finally get this out there, but I'm sure you were sitting there when some of these comments were coming in like, oh my God.
Well, let me give you the story of the paper and then ask the same kind of questions. The paper began about a year ago when the Alzheimer's Association asked me to do a summary talk at the end of the day of a conference in Washington. Okay. It was on animal models for Alzheimer's. And there are two adages that come to mind with regard to Alzheimer's. One is, quote, everything works in mice, nothing works in humans. The answer is why. The other one is Alzheimer's is the graveyard of companies. Okay, why? Same kind of question. And so I gave the summary talk, and I said, we have two problems in this field. The first is, what we have in this field is we have targets and we have techniques. We have targets, for example, beta amyloid. We have techniques, for example, monoclonal antibodies, and they're very good. What we entirely lack, however, is an overall map of what we're doing. It's like blindfolding ourselves in the dark and throwing random darts around the pub and hoping we hit the bullseye. The dart's fine, your throw is fine, but you don't know where you're throwing. And that's part of the problem. So I said, what we lack is an overall model, and the model not only has to be able to account for Alzheimer's and all the data, the risk factors, the downstream but it also has to account for other age-related dementias, and it has to account for cognitive decline in animals. I had this one wonderful slide where I had mice, rats, dogs, cats, rhesus monkeys, and I suddenly paused and I said, I just realized that in my entire career, I've been bitten by all these species, including people, and I don't just mean kids. They've all bitten me, and they have, including a couple of rhesus bites on my body, and a gorilla bite. But the point is you have to account for all these. If you're going to have a model for Alzheimer's, it's got to be able to do more than that. It's got to be able to account for all of these, and how does aging play a role? And afterwards, I got mobbed by people, which I'm not used to, particularly complimentary mobs, who came up and said, yes, yes, we agree, totally, you're right. And one person said more thoughtfully, he said, you told us we need a model, and I completely agree. Do you have a model? I said, yes, I do. I didn't want to impose on everyone else. I just want to point out we need one. That's a critical thing. He said, will you write your model up? And I said, who are you? He said, well, I'm actually editor-in-chief of Alzheimer's and Dementia. And then he asked to join our scientific advisory board, which tickled me no end. So I wrote the paper up, and then I've never had academic editors take me out to dinner before on their dime. And they'll look at the paper. It took a while to put it together. And when I did, it was about 3 times the length of a normal paper. And initially, it was 17,000 words, 269 citations, 8 figures, which it roughly is now. And when I got done, we had run it through 8 reviewers. And I expect 8 reviewers, you're going to get at least half of them are going to say no. And what surprised me was that pretty much all 8 reviewers were complimentary. One was a bit on the snarky side, but also complimentary, which is odd. And they had questions, but they liked the idea of having a model. And 6 of the 8 reviewers said, we think the paper is too long, which I agree, but we'd also like you to add more about our particular question. And then the editors came to me and they said, listen, we're going to just publish the whole thing, but we have one more question, one more thing to ask of you. And I thought, oh my God, what? And they said, what we'd like you to do is write an extra 1,000 words on the dialogue between you and the 8 reviewers. What were the questions that they asked? What were the things they wanted more clarity on? What were the things that brought to mind their issues? And so that's where that epilogue came from. So it ended up being about 18,000 words and now down to 250 references and still the 8 figures because some of the references were duplicates, cited several times. And that's the way it came out. And what surprised me was, I have about 100 publications, I think, in my lifetime, which is odd because I never wanted tenure or anything else. I just wrote things that intrigued me. But I've had reprint requests before. People write in, a couple of JAMA papers I wrote, people write in and they want a reprint, a dozen here, a dozen there. We had more than 500 reprint requests in the first 2 weeks. I've never seen anything like that. And I had a dozen and a half requests to come give talks around the world, to which my response was, nope, I'm busy, thanks. And about a dozen inquiries from venture capital groups. It seems to have struck a chord. And there are 2 ways to look at this. One is, yes, we need a model. It doesn't mean my model is right, but we do need a model. I think everybody agrees with that. We need a model that gives you a uniform field that accounts for the data consistently and offers some potential innovative points of intervention. I think most of us in the world agree on that one. The other question is my model right? And again, I think it is, but we can take it to clinical testing. And as I always say, 1, that's the point of any scientific model. If you can't test it, it's not science. And 2, theory and models are good. Data beats them every time.
Absolutely. And it's a great paper. I mean, it's really, I had to go through it a few times, but it's.
I had to go through it several times too. I still don't understand my own paper. So really.
I said, well, I have one other question for you before I get to the wrap up. And I just wanted to add, if this is something that's sort of outside the scope, then we can just avoid it. But based on your telomere expertise, when you were at the Michigan State, there was a paper in the early 2000s published by evolutionary theorist Brett Weinstein called the reserve capacity hypothesis, which focused on this somewhat controversial theme that a certain group of hyper elongated telomere animals at the Jackson laboratories, based on the way they were kept and nurtured and so forth, and based on the fact that a lot of the drug testing models came out of there, skewed a little bit in the sense that because they had much longer telomeres than a lot of the other mice you find, the the effects of certain drugs in toxicology testing and so forth, heart disease, liver damage, and so forth, were skewed. It was one of those things that was kind of quiet. You Google it, you see a little discussion here and there on it, but you didn't hear much more. In your books, have you come up, did you ever, I apologize, I haven't read all your stuff, but did you ever focus on this issue? And I don't know if it's a non-issue nowadays, but is this something you ever came across in your writing as something in sort of the pharmacotherapeutic development path?
Yeah, there are any number of problems in dealing with telomeres.
Right.
You see these misconceptions right and left. The first thing, let me say, as I've said already, telomeres don't cause aging.
Right.
They don't. They play a role in it. Secondly, something I've reiterated now for 25 years and people still don't get it. Telomere length is unimportant. Relative telomere length is important.
Yeah.
And if you plot out telomere length, you find it's all over the landscape. For example, there are varieties of mice with telomeres that are literally 10 times longer than mine, have lifespans 40 times shorter than mine.
Right.
It doesn't matter. But if you look at the rate of telomere loss, you find it maps out very nicely. Mice lose telomeres fast. I lose telomeres relatively slowly. Humpback whales, it depends on the species. And furthermore, it's not just the rate of telomere loss, that is the relative length as opposed to the absolute length. It's really the changes that induce in gene expression and what that does to, for example, recycling of molecular pools, for example, DNA repair enzymes, and mitochondrial function, and lipids, and I mean, it just goes on and on.
Right.
The other thing that I see that happens all the time is one, in most cells, when you look, it's not the mean telomere length, it's the shortest telomere length that sends the pace. And people find it very easy technically to measure mean telomere lengths, but that's not the right thing. That's like measuring your gray hair to try to figure out whether you have osteoarthritis. Yeah, there's a relationship, but that's not the thing you're supposed to be measuring. You should be measuring, or they'll measure the wrong cells, which are much the same sort of issue. They'll measure peripheral leukocytes and they'll infer something about glial cell function or chondrocyte function or podocyte function. No, if you're going to comment on chondrocytes, you should be measuring chondrocytes, not circulating lymphocytes. The other problem is that they'll measure peripheral lymphocytes and they won't account for the fact that, again, that's a dynamic population.
Right.
Right. So if right now, let's say that your mean telomere length or shortest telomere length, we say 9 kilobase pairs right now. But now tomorrow, your lover leaves you, you get fired by your job, somebody runs over your dog. You end up with a viral illness, you name it. Your doctor calls to tell you you've got terminal cancer. Everything goes wrong in life at once. If I come back in a week and measure your telomeres, they will look god-awful. Now, if it turns out all those things get fixed, and we put you on a vegetarian diet, and we have you meditate, and we take away all your stress, and we make you happy, we take you to Disney World, your telomeres will be longer. But it is not because they got longer, it's because the peripheral churning has now slowed down. And so you're sort of back to your baseline. That sort of mistake gets made all the time in the literature. People will use some sort of intervention, as I say, to put you on a vegetarian diet, have you meditate, and they'll claim they made you younger. Well, maybe you did. That's not what the data showed. Right. But those are the kind of problems that are throughout here. People think that telomere length is important. No, relative telomere length. They're looking at the wrong cells, they're looking at churning, they're using the wrong measurements. So most of the data doesn't mean much.
I appreciate that. That's a nice overview and sort of lay of how one should go into this space in terms of their own research and what they should be looking at. So I appreciate that. Going back to Mike Fossel, we typically ask a wrap-up question about sort of important influencers throughout your career. No doubt you had a fascinating one. You've met a lot of fascinating people in academia and industry and in government. Any specific folks that you might want to give a shout out to at this point that were really instrumental in keeping Mike Fossel on this particular path, that if it wasn't for them, I'd be talking to Mike Fossel, lawyer or real estate tycoon or something else, as opposed to what you're doing now?
Yeah, a lot of people. And as a sort of a joking human interest aside, I'll say Coco the gorilla, because I got to babysit her for a year.
Is that the one that bit you?
Yeah. I'm probably the only person you'll ever met who has bitten a gorilla, been bitten by a gorilla, kissed a gorilla, wrestled with a gorilla, and been,
I won't do any of those things. But that's okay.
So, I'm all in favor of Coco. I will miss her forever. let me point out one more person.
Okay.
And just because it's got a deeply ironic note for this conversation.
Sure.
And that is the chief scientist at Geron Corporation 25 years ago, I think I met him now almost 30 years ago, was Cal Harley.
Okay.
Cal Harley probably knew more about how telomeres and telomerase work in a really effective way than anyone on this planet. He used to say that I know more about telomeres, telomerase, and aging than anyone on the planet. Doesn't mean either one of us are right, but it's kind of nice. So I knew Cal well back in the mid-90s. In fact, he and I were working together on a biotech company about 7 or 8 years ago. And both of us have gotten intrigued by Alzheimer's and the role that telomeres play. I like Cal a ton. I had enormous respect for him. His wife gave me a call about 3 weeks ago to tell me that Cal has Alzheimer's. And I just went out to visit them and spent 3 hours holding rocks with Cal. The irony in there is just as deep as it gets. So here's off to Cal.
I appreciate that. And that's
I wish it weren't so.
yeah.
And I also wish I could move faster so I can do something about it.
Absolutely. Absolutely. Mike, it's really it's been an honor having you today, talking about your history and everything that you're doing now, and really pushing forward what I'll say, these new narratives, these approaches that we've had the blinders on way too long, in my opinion, in regard to this and other therapeutic spaces, specifically this one, because it is such a, what we call the silver tsunami, or whatever you want to term it. It's coming fast and furious, and we see groups like Pfizer with their $60 billion or so just running away from this disease. It's good to know that there's minds like yours out there that are really looking at the alternatives and what we need to be, where we need to be, what we need to be doing. And so I really appreciate everything that you're doing, and if I could take time out of your schedule to come talk on the show. For everybody that's going to be watching on the YouTube channel or listening, when speaking Dr. Michael Fossel, founder and president of Telocyte, pick up his recent paper, A Unified Model of Dementias, Age-Related Neurodegeneration. Really an amazing synopsis of the space, what's going on, what's worked, what hasn't, and where we potentially going to find those solutions for this tremendous area of unmet medical need. Mike, thanks for what you do, as we say on the show, for moving the human story forward, moving the Alzheimer's story hopefully in reverse. Any last words? Take us out if you would. But really, thank you for coming on the show.
No, just that it's, I think we can cure Alzheimer's and other age-related dementias, but we cannot do it by being naive and having magical thinking about small molecules. We've got to actually understand the disease. Once you do that, here's again the irony. If you think it's simple, it's just a matter of things falling apart, you will never be able to solve it. If you understand how deeply, enormously complicated it is, then treating it turns out to be simple. Well, conceptually, technically still difficult, but doable.
Yeah, I'm glad you're focusing on it, though. Mike Fossel, thanks so much for joining us.
Thanks, Ira.